Theme is the new bright flow cytometry panel design with Invitrogen NovaFluor Dyes. I'm Dr. Anson Blanks of Thermo Fisher Scientific, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Thermo Fisher Scientific. To learn more, please visit thermofisher.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type your questions into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. This webinar is educational and thus offers free continuing education credits. Please click on the Continuing Education window at the bottom of your screen to obtain your credits. I'd like to now welcome our speaker Kathryn Friend, Business Development, NovaFluor Dyes at Thermo Fisher Scientific.
Hi there, thank you very much. The title of my talk today is Clean as the New Bright. So today I'm going to be looking through and talking about flow cytometry panel design, in particular with using the Invitrogen NovaFluor Dyes. And we'll be looking also at utilizing the Thermo Fisher panel builder as well to incorporate NovaFluor Dyes into the panels. So if we're looking at optimal panel design, particularly if we're looking at it from a fluorophore perspective, we really need to look at, say, 4 different considerations. So the first is spectral overlap, where you have to look at the fluorescence spectra itself and see where it overlaps into other detectors. The second is antigen density, where we're looking at how many antigens are there either on the cell surface, how many antigens of the marker of interest is actually there within your sample, and therefore this will really play a big part as to which fluorophore you assign to that particular antigen. The third is spillover and spreading, and obviously every fluorophore has some kind of level of spillover and spreading, and we need to take that into consideration. And the fourth is stain or separation index, and this obviously needs to be a consideration because it affects the brightness or the brightness of, it really shows the brightness of the fluorophore, and that again is really important in terms of panel design and being able to balance your panels properly by matching your antigen density with your fluorophore brightness. So I'm going to go into each one of these, and then I'm going to go and look at NovaFluors and how they can be incorporated into your current panels and how you can create new panels with NovaFluors. So let's look at spectral overlap. So this occurs when a fluorophore's fluorescence, it spills over into the detection of another fluorophore. So there are many fluorophores. In fact, most fluorophores, all fluorophores have some kind of spillover. Many of them have quite a broad emission, or they're excited by multiple lasers. And each one of these causes spectral overlap and needs to be considered in your panel design. Antigen density, and in fact I'll go into a little bit more detail with antigen density than spectral overlap at the moment. So antigen density refers to the number of target molecules expressed on a given cell. So your antigen density really needs to be considered in every single panel design. And in fact, this is one of the tricky things that I have encountered in making panels. The reason is that antigen density can vary depending on your cell type, your disease condition, your drug treatment, or whatever treatment you happen to be putting onto your samples, your sample preparation, and your certain activation states of your cells. And so these need to be, again, considered in terms of panel design. The best way that we've found to be able to determine antigen density, apart from testing it yourself, is to review your recent available literature regarding your cell type and your antigen of interest. And again, if that's not available, you're going to have to test them yourself. The other way to look to kind of determine antigen density without testing it yourself is to look at other other available resources. There's two that I can suggest at the moment that are really quite helpful. OMIPs, or Optimized Multicolor Multi-Immunofluorescence Panels, are special peer-reviewed cytometry Part A open access publications. And they basically outline a number of different panels that have been designed and tested and work. And so they're a really good resource if you're wanting some place to start. There's also city maps. City maps that are available. I've given an article, a website here that's available from the HCDM. It basically maps the expression or the general expression of human CD1 to 100 on particular immune cell subsets. And so these are really quite interesting. Some of the other companies tend to have a couple of documents on antigen density, but I found that these, again, because this varies substantially on cell type and sample preparation, these aren't always exhaustive, and so I would always use a number of different factors to determine the antigen density of your particular sample type. So let's talk about antigen density though, because that really affects how you set up your panel. Okay, so let's start with these low antigen expression level markers. These ones who are low or you don't even know what their expression is like. Oftentimes, for these markers, you can see if their antigen density is low, or minimal based on the amount of fluorophores that are available in the market on these particular markers. I'll also show a little some data later where you can actually use the market available information to really determine whether your antigen will determine what your antigen density is going to possibly be like in your sample. But either way, these low antigen density markers really have poor expression. Their populations may be smaller, but what you need to do is save your brightest fluorophores for these. And I would always assign these first. You want to make sure that in any given panel, you're actually looking at these particular markers because they're often the critical target in the panel. Next, what I would also look at is these high antigen expression levels, the high antigen density markers. And these are the ones that I'm actually, in panel design, are assigning a lot of the NovaFluors to, because NovaFluors, depending on the NovaFluor, can be quite dim. And so in a panel design, what you want to be doing is you want to be balancing out your antigen levels with your fluorophore brightness. You don't want a whole stack of bright fluors because that is going to, because your antigen levels will vary. And so you really need to be able to match these. So again, I would find that these high antigen expression level markers, they're often well characterized. They usually can identify quite broad subsets of cells. They're usually your lineage markers. The expression is either positive or negative, so really easily defined, and they can often be used as the parent gate. And so you would assign these to the dimmer fluorophores, or the targets that are most affected by the spillover. So after you've done that, so you've assigned your low antigen expression markers to your really bright fluorophores. You've assigned your really high antigen expression markers to your really low fluorophores. Then you've got everything else. And then you just need to, again, match up your expression levels with fluorophore brightness. And that's more of, and a lot of times it's also about what's available by that stage as well. So, it's a kind of this is, filling in the gaps as well in your panel. So, as I said beforehand, you can use what's available in the marketplace from all these different companies to actually determine the level of antigen brightness that you need to kind of look at the antigen brightness of a particular marker on a particular cell type, or the relative one. So for instance, this is CD4, and as you can see, this CD4 conjugates can have three different levels with these three different conjugates. You can have three different levels of separation index depending on the fluorophore that it's attached to. And so you'll have, for instance, if you're using CD4 happens to be in this sample quite well expressed. If you're using a really high brightness fluorophore like APC, you'll see a really big shift over here. So really big difference in between your positive and negative cell population. Whereas, there's my dog in the background, this is working from home. Whereas if, for instance, you're using a less bright fluor, for instance, like eFluor 450, you will see a smaller shift between your positive and negative cells. Fortunately for CD4, this won't matter too much. You can still see quite a good separation between your positive and negative cells. And I would suggest if you're looking to have a look at antigen density, and you see that, for instance, lower brightness fluorophores don't necessarily separate out your positive and negative cell populations, then I would suggest that this is a really good way of determining whether or not you should be using a brighter fluor on those particular markers, as well as obviously going back to your research on the current publications as well. So how we determine brightness, there's two ways that we're used in the market at the moment. You either use stain index or separation index. At the moment with the NovaFluors, the way that they're set up, we actually use separation index because it's providing just a slightly more accurate measure of fluorophore background compared to the staining index. Both of them will just consider the difference between the positive and negative populations, and what we're trying to define here is just a relative level of fluorophore brightness between different fluorophores. So we use that with our NovaFluor panel design to be able to determine the level of brightness of the NovaFluors as compared to many of the fluorophores that are out there in the marketplace. And you'll see that on the NovaFluor landing page on the Thermo Fisher website. So another consideration that we have to, take into consideration is spillover and spreading. So many fluorophores either have this broad emission spectra. So for instance, here we can see the brilliant violet 510 is emitting over a number of different detectors, or you have these cross-laser excitation, for instance, with PE and the PE tandems, where these dyes are excited by a number of different lasers. Both of these particular anomalies, I would say, for fluorophores will affect the resolution of the entire panel, because what they're doing is they're spilling over or spreading into other detectors or other channels. And as you can see here from Laura Johnson's blog, so the APC Cy7 is spilling over into the PE Cy7, and will need to be compensated to determine the real value of the marker that's determined in the PE Cy7 channel. This kind of leads me to the discussion about the co-expression of markers. And so spillover spreading, particularly for co-expressed markers, is quite a problem because you won't be able to determine the difference, the real value of these markers. And so you want to minimize the spillover of, say, your highly expressed population into detectors that are a little bit more sensitive, or those that have lower expressed markers. The way you can do this is you need to space out your fluorophores of co-expressed markers across your panel. And if you can place mutually exclusive markers in adjacent channels, that's kind of the best option for you as well. Another way to do this is to use a fluorophore that has a very slim spectra and doesn't spill over across a number of different detectors to reduce the spillover impact on the resolution of this biology. And so this is what we're trying to do with NovaFluors, is reduce the spillover spreading and so increase your biological resolution. So if we look at panel design and the requirements that you need for panel design, first of all, you need to understand your targets of interest. So you need to really understand, first of all, your sample type, and obviously all of the conditions that your sample is under, the markers of interest, and again, I reiterate that marker abundance or antigen density is really important as well. In terms of strategies, you need to understand your cytometer configuration and also your fluorophore compatibility. You really need to understand how fluorophores work with that. You need to have a good gating strategy, and this will determine whether or not you put certain markers in adjacent channels. And then obviously you need to look at marker co-expression. Again, this will determine whether or not you put things next to each other in your panel. Obviously online resources will help to build your panel. And so this kind of brings me to introducing NovaFluors. So NovaFluors are like a unique set of dyes. They have unique spectral signatures. They're designed with this narrow emission spectra in mind. They're particularly engineered with minimal cross-laser excitation and to decrease the spillover spread across the spectra to increase the resolution of your panels. They're actually easy to add to existing panels and easy to create panels with NovaFluors, and again I will go through that at the moment. And they can be used for conventional cytometers and for spectral cytometers as well. So let's look at NovaFluors. What are they? So they're actually engineered single-strand DNA dyes. So what we do is we engineer these single-stranded DNA with a number of fluorophores embedded in that DNA. And so then they anneal into this Phyton structure in the middle here. And this Phyton structure holds these particular fluorophores in place. And so what this means is these fluorophores fret off each other, producing a specific emission spectra. They're then attached with an oligo label to your antibody here. And so what we can do, in fact, this is kind of unique, is that we can add extra fluorophores if needs be, and we can actually increase the brightness. And so here on the right-hand side, you can see the first one up the top has two labeled Phyton, or has two arms that are labeled, and there's one down the bottom has 4 arms that are labeled with Phytons, and this can actually increase the brightness. And so what we have done is that we've created 2 different, for instance, we have created 2 different dyes that emit in the same channel but have different brightnesses as a result of this engineering of the brightness we are able to do. What's nice about the NovaFluors is that using this particular structure, we have now managed to release 19 NovaFluors over the last year. And each one of these are either set to compete with a dye that is particularly spectrally promiscuous, if I'm allowed to use that word, in fact, so that it spreads across a number of different detectors, or that it fills in the spectral space. So, there is a lot of, there's a lot more detectors at the moment with these current cytometers than there is dyes, and so again, we're starting to fill in some of the spectral space that is available at the moment using NovaFluors. And so the nice thing about NovaFluors is that the nomenclature is really easy. You basically can see where it emits based on its name. So NovaFluor Blue 510, for instance, emits at 510 off the blue laser. Example. The other thing that we've done with NovaFluor dyes is that we've really tried to engineer them so that they have quite a slim spectrum, so that they actually can be used in adjacent detectors. And as you can see here, these are 2 dyes, NovaFluor Blue 530 and NovaFluor Blue 510. That's in a spectral cytometer, you're actually able to determine both of them. In a conventional cytometer, maybe not, but because their spectra is different, you're actually able to use them together. And they have like minimal cross laser excitation, so you can see that they you can use them together in a panel. What this means is that you can increase the capacity of your panel to be able to add more markers. So I did mention beforehand that you can engineer the brightness, and what we've done in terms of the nomenclature is that we've looked at separation indices, comparing CD4 across a number of different fluorophores on an Aurora, a Cytometry Aurora, and we found that the best way to determine or to name our different fluorophores in terms of brightness is to just put this separation indices kind of figure next to it. And so, again, this is the relative score, but we found that, for instance, for our NovaFluor Blue 610 dye, we have one dye that has a separation indices of 30 and another dye that has a separation indices of about 70. So we have named them NovaFluor Blue 610 30S and 70S. And again, what this means is, for instance, if somebody is doing a longitudinal study and they want to test for a particular marker that changes in terms of density across that longitudinal study, you can swap out one fluor for another in your panel, depending on antigen density. So, which is quite handy, I think. Another kind of anomaly with NovaFluor dyes is that we've found that they can non-specifically bind to monocytes and macrophages in a similar way to other dyes like your PE and APC Tandem Dyes do. And so we recommend, we recommend that for every NovaFluor dye, you use the cell blocks monocyte and macrophage blocking buffer. It can be used instead of other macrophage and monocyte blocking buffers that are on the market at the moment, and can also stop the non-specific binding of your PE and APC tandems to macrophages and monocytes as well. And we recommend adding them to, say, the antibody mixture just before you add your cells. So it's just something that we found, and we include some cell blocks with every single NovaFluor dye that's sold at the moment. Another kind of anomaly for NovaFluor dyes is that obviously they're DNA based. So you can't use anything that is a DNA binding dye for viability. So that includes, say, your DAPI or your 7-AAD or pyridiniamide dye. We do recommend using amine reactive viability dyes such as the Invitrogen live dead fixable dead cell stains. So what we have available to us is we've got a list of directly conjugated lineage markers conjugated to all 19 NovaFluor dyes. We also have a conjugation kit, which is really handy if you wanted to add an extra marker to your panel and you have no space to put it. We've got 19 different NovaFluors that you can slot in wherever you need it to. And so this conjugation kit has a 3 easy step process. You basically attach your single-stranded DNA NovaFluor linker to the antibody, then you remove your excess linker and attach the NovaFluor dye, and then you anneal the NovaFluor dye together. It's a really easy 3-step process, should take 1 to 2 days to do and not much hands-on time. So if you need to use this, we would highly recommend this antibody conjugation kit. So let's look at adding markers to a panel, and this is the considerations that you need to take, that you need to look at to minimize the loss of resolution and maximizing your data integrity. So in any panel design, if you add another marker to the panel, this is really going to increase your complexity of the panel and therefore decrease the possibility of the resolution of your biology. You really need to know your instrument detectors and understand your dye spectra. It's also really important to match your fluorophore brightness with your antigen expression level, and obviously, you know, that becomes much more important as you increase the number of number of markers in your panel. A really well-balanced panel in terms of antigen and fluorophore brightness makes such a difference to the resolution of your panel. So, what do NovaFluors do? Well, we actually have a number of them that can compare with the current fluorophores that are on the marketplace at the moment. And if you have a look, these are all the NovaFluor blue and yellow dyes that are competing with normal traditional dyes like FitC or PE or APC, for instance. One caveat I would say is that the brightness is not always the same in terms of NovaFluors, but then brighter is not always better. Cleaner is better than bright at the moment if you're looking at panel design. And so when you are replacing your traditional dyes with NovaFluor dyes, I would really consider the antigen density and the brightness as well with your panel. So let's look into that a little bit more. For instance, so your PE and PE tandem dyes, they're excited by a number of different lasers. And so what this means is a spectral spillover across a number of different laser lines. What we can do is that we can look at replacing this PE tandem, which is PE dazzle, with something that is spectrally slimmer and isn't excited by 2 different or 3 different laser lines. So what we've done here is, instead of using PE dazzle, we've actually replaced it with not 1 but 2 NovaFluors. So here you can replace PE dazzle with NovaFluor blue 610 and NovaFluor yellow 610, which is spectrally slimmer. And so what this means is you can get a 2-for-1 upgrade of your panel. The caveat is that PE dazzle is really quite bright, and neither one of these fluors are as bright as PE dazzle, so you do need to look at antigen density and where that will work in your panel to see if you can do this as well. But in saying that, it's an easy way of upgrading your panel and adding extra markers if you wanted to. Plus, reducing the spillover spread from PE dazzle is also handy as well. So let's look at how we've done that. So for instance, here we have a traditional panel on an Aurora, sorry, not an Aurora, my apologies, a traditional panel run on an Attune. And here we have like 11 different colors. And if you have a look here at BL2 and YL2, these 2 detectors can't be used because BL2 is currently experiencing a whole stack of spillover spread from the YL2 detect channel here. So the PE dazzle 594 with CD45RA is spilling over into BL2, so it can't be used. Conversely, the YL3 PE 5.5 possibly could be used in this detector. However, it is receiving a whole heap of spillover spread from the BL3 channel here. So from per se PE 5.5 is spilling over and spreading into YL3. So we're not using a particular marker in there because that would affect the resolution. What we can do is that we can replace these. We can replace per se PE 5.5 and PE dazzle 594 with NovaFluors. And by doing that, we actually open up 2 extra detectors to be able to use them. So what we've done is we've added an extra couple of markers in here. So as well as replacing kind of directly replacement dyes, and so for instance, AlexaFluor 488 can be replaced by NovaFluor Blue 530. We've actually added, we've replaced those dyes, we've also added an extra couple of those as well. So we've added CXCR4 and CD28 into this panel as well. Now what does that do? It gives us a little bit more resolution and greater depth. So in fact now we're actually better able to resolve our naive T cells, our central memory T cells, and our effector memory T cells as a result of adding these extra 2 markers to your panel. What we've also done though, and this is really quite subtle, but really important, and it actually becomes more important the more markers you have, or the bigger the panel is that I know this is quite small, you probably can't see it that well, but across the spectra, by taking PE dazzle out of this particular panel, right across the spectra, we have reduced a whole heap of spillover across a number of different detectors. Ideally, what this will do is increase the resolution of those particular markers in each one of those detectors. So by taking PE out of this particular panel, we have increased the resolution of the entire panel. And so this is again what we're trying to show here with NovaFluors, is that they can be used to increase your biological resolution of your panel. So optimizing panel design. One really good way of looking at your panels is to look at your spillover spread matrix after you've run it, say, on something like FlowJo, and you can identify, using the spillover spread matrix, those dyes that are adding most to spread across your panel. These ones are the ones that we'd probably be wanting to take out of your current panel and replace with something slimmer in terms of spectra, like a NovaFluor dye. You want to also identify the available detectors that you've got available to you. So oftentimes you will have, particularly with these NovaFluor dyes that are able to slot into spectral spaces that didn't have any other dyes in them beforehand. Identify those available detectors, and then you can see what else you can add to the panel. You want to replace these high spreading dyes that obviously you've identified in your SSM analysis with your NovaFluor dyes, or any other alternative dye that has less spreading than these high spreading dyes. But again, the caveat is that you want to consider quite carefully the level of antigen density in replacing these dyes. Not every dye is the same, and you need to consider this quite carefully. What you could do, though, is either replace it directly with the NovaFluor, or you can swap your markers around again to balance the antigen expression with dye brightness for these panel designs. So next, I want to look at building a panel from scratch, and for this, I wanted to look at the panel builder, the Invitrogen flow cytometry panel builder that's available online at the Thermo Fisher website. So I've used a number of different panel builders. In fact, I think I've used most of them that are on the marketplace, and I really think, I mean, I'm not being biased here, but I really think that this is the best one, and I'll tell you why. What it does is it allows you to add any kind of configuration of your cytometer, and the nice thing about it is that it will list the fluorophores list the products in terms of list the antibodies that you're putting in there in terms of product availability, and so it'll list the ones that have less available direct conjugates first, which is the ones that you want to assign because they're the ones that you can never find any available direct conjugates to. So I think this is a really great panel design tool to do this. So what do we do first? Going into the panel builder, first of all, you can select your cytometer, and we've got hopefully we're listing most, if not all, of the cytometers that are on the market at the moment. Alternatively, you can actually customize your cytometer setting using the tab down the bottom there. So first of all, you need to just sit there and set up the panel builder to your flow cytometer configurations. And the settings can be saved, and there is, again, an extensive list of conventional or spectral instruments that's on there at the moment. You can use this to change or modify the cytometer information, and you need to make sure that the filter settings on the particular cytometer that you've chosen actually match those that you have that you're going to use. So then the next step really is to select your antibodies, your proteins, and reagents. So initially, like, this is the next step of the panel builder. First of all, you can put in the antibodies that you already have in your panel. The next step is to add any fluorescent proteins, things like GFP or RFP or any other proteins that you're using within the panel. Then the next step, obviously one of the most important, is to add all the markers in that you need. And for this, it'll actually show you can add in the antibodies, and you can add in the antigen density as well of these antibodies, which is really quite handy. And then the final step is to add in any extra non-antibody reagents, things like your cell viability of your protein, your functional protein dyes, or your cell cycle analysis dyes and other things like that. Next, again, the view from this point on depends on whether you've chosen a conventional cytometer or a spectral cytometer. They have different views. So first I'm going to talk about conventional cytometers. To start off with, you have this, once you've got everything in there, including all of your different markers, it'll show you this matrix view of your columns, your channels up the top here, and your antigens listed along the rows. Each one of these coloured dots will indicate a particular fluorochrome that is available in each particular channel. And so each one of these basically indicates a product that is available in a particular fluorochrome that can be detected within that detector. And the nice thing about it, though, is that, the black flags will indicate your recommended channels, and these will decrease depending on how many you choose. And as you go through and highlight each one of those, highlight the particular product that you're going to be using, these will grey out, and so you can't then pick something that will be detected in the same detector. So the recommendations, the black flags for these will be based on, first of all, your protein abundance from your antigen slider and the fluorochrome abundance, and they will look at the antigen brightness and the antigen density and the fluorochrome brightness in terms of being able to recommend the right product for that particular marker. In a spectral cytometer, you have a different view, and so here you still see the channels and the antigens in this matrix view, but you also get this kind of thing, this complexity score. And so as you're going through picking your fluorochrome, this is using a drop-down menu, it'll increase, the complexity will increase. And so maybe I'll show a bit better on the next page. If you pick a fluorophore that is going to compete directly with another fluor, or it increases the complexity incredibly, you will get this kind of orange flag. And so I would always consider about suggesting another fluorophore. And as you see, when you're picking these fluorophores, you need to just there's a drop-down menu. And what's nice about this is it'll show a complexity score for the addition of each of these of each fluorophore that you've got available to you. And so it'll also show in the top right corner a combined complexity score. And so obviously you want to make sure the complexity of your panel is as minimal as possible, because this will add to the, a lower complexity score will increase your resolution. Final selection is when you finally decide on the antigen that you want. You basically click on it, and you can see a whole heap of detailed information on that particular antigen that's available, or sorry, that particular product that's available as well. The nice thing about it then is once you've selected your clone, you can have a look at it in a bit more detail as well. The final point of your of the Thermo Fisher panel builder is that you can then export any of these lists of the antibody conjugates. And again, this is what's really this is another really nice feature. You can export it as a spreadsheet or a PDF, so you can order it straight away. Some people can order it directly in your cart if you wanted to, or you can again give this spreadsheet to your purchasing officer to order it straight away. And the final panel will include all the selected products, the placeholder reagents, and any other reagents that you want. And plus, for the conventional cytometer, you'll also see this spectral viewer tool, which will allow you to see really the spectral spillover for each one of these fluorophores that you've picked. So, in terms of panel design with fluorophore dyes, what I have been doing is putting all the putting all the markers into the flow cytometry panel builder. We've identified all the markers with minimal available conjugates, and as I said, what's nice about the panel builder is that these will be listed from top down. So those that only have 3 available conjugates will be listed at the top, and those with 12 available conjugates will be listed towards the bottom. This is good because you can assign these first to things that are available. Then I would always, again, identify these markers with these low expression levels. And again, you'll know that from doing a lot of your reading with all of your, to determine your expression levels of each markers within your cell type. I'll always assign these to the bright fluorophores first. And then the nice thing about this is then I would add the NovaFluor dyes, particularly on lineage markers, because that's what's available right now as direct conjugates, and then match the remaining markers with the available fluorophores depending on antigen density and brightness. It is a bit of work in progress, but this is certainly the way that we can create a panel with NovaFluor dyes, and we're happy to help you with this as well. So, in summary, the question is, what can you do with NovaFluor dyes? And really, there's 2 things that we can do. And I've showed that in this presentation. First is that we can actually do a panel upgrade. We can add additional markers, or we can take some of those troublesome fluorophores out of the panel and replace it with a fluorophore with a slimmer spectra to reduce the complexity of these panels. We can do that with panel upgrades. What we can also do is that we can look at broader or deeper immunophenotyping. And I'd like to point out there is another LabRoots presentation that you can see that was given by Seddon Thomas last month on the good and bad and the ugly of 45 color panel design, where she goes into talking about higher dimensional flow cytometry and what works and what doesn't work, and what's best to maintain your biological resolution. So I would recommend going and finding that if you're interested in broader or deeper immunophenotyping. Finally, this is my summary. I reiterate, clean is the new bright. For a long time, brighter is better has been the mantra for panel design. As you increase the amount of markers in your flow panels, this is not holding true at all. In fact, brighter will work against you if you're looking at brighter fluors on high antigen density markers. And so you really need to be able to balance your panel and balance them with a cleaner spectral, a fluorophore with a cleaner spectra. And this is what NovaFluors are able to offer you. They're able to increase the size of these flow panels and decrease the spillover and spreading, which also then increases the biological resolution. And with that, I'd like to finish my talk, and I'd love to hear some questions from the audience if you have any. Thank you very much.
Thank you, Kathryn, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen, and we'll answer as many of your questions as we have time for. Let's get started. Our first question is, will there be any issues when using DNA viability markers and NovaFluors?
Thanks. Good question. I'm not sure that we addressed it earlier, but DNA viability markers will have an issue, yes. These are DNA-based products, and so any kind of intercalating dyes that are going to bind to DNA are going to react with the NovaFluors. And so we do not recommend using anything like your propylium iodide, for instance, or any other intercalating dyes. What we do recommend is using an amine-based live dead stain, for instance, like your Invitrogen live dead dyes. These are the best ones to use with NovaFluors.
So a question that we get pretty often, Kathryn, what is the size of the Phyton?
Good question. Thanks, Anson. So the Phyton itself is actually a really stable structure. It is about 20 kilodaltons in across and about 20 nanometers across and 150 kilodaltons in size. So the size is about the same as your antibody. And so one thing that, again, I didn't address is that for all of our direct conjugates, we can actually guarantee that there is a 1-to-1 Phyton to antibody ratio, which means actually you can use these fluorophores to quantify the amount of antigens on your cell surface. And I'm pretty sure there's not many fluorophores that are quantifiable as well that are around there at the moment. So it's kind of an extra added bonus for the NovaFluors. The other thing that is interesting about the structure is that it holds, because it's a DNA structure, it actually holds the fluorophores in place. And in fact, it's hydrophobic, so it actually stabilizes the fluorophores. What this means is it's incredibly stable, both at room temperature and in certain fixatives. So it can be used, can be sent to Australia, for instance, at room temperature for 2 weeks, and also it can be used with a number of different fixatives. And we're investigating these further as well.
So again, we want to encourage questions from the audience. You can just write your question right in the Q&A box. Going along the lines of what you just said about stability, so I think there's a lot of documented evidence about the stability issues that some tandem dyes have. With these NovaFluor dyes, are they tandem dyes? Can you talk a little bit more about that?
Good question, Anson. NovaFluor dyes aren't tandem dyes. So by the definition of tandem dyes, they're things like PE Cy7 or APC Cy7, where they're actually held together by a covalent bond. Conversely, and then they use these fretting technologies the fretting technology to be able to emit in a different detector than the primary fluorophore. NovaFluors use the fretting technology, so they utilize a combination of different dyes, and they spread off each other to be able to emit in one particular predefined detector. But because they're actually held in this DNA structure quite rigidly, they don't have the same kind of issues that tandem dyes have in terms of stability. You know, I'm pretty sure everyone's had a problem with APC Cy7, where the Cy7 drops off because the covalent bond doesn't last very long in fixative solutions, or doesn't last very long over a long period of time. You don't have those issues with NovaFluors. They're actually highly stable and they don't drop off. And so this means that you can use them over longitudinal studies. You can also use them again in a number of different fixatives and buffers, and we have tied a number of different ones as well.
Awesome. So we've definitely tested the NovaFluors and paraformaldehyde as well as formalin, and we have not seen any effect on fluorescence, so that's pretty cool. Question from the chat: can I use NovaFluor dyes on seismic Cube 8 flow cytometer?
Yes, you can use NovaFluor dyes on any flow cytometer, so long as they have particular detectors that would be able to detect the NovaFluor dyes. And I actually don't know the configuration of a seismic Cube 8 flow cytometer, but I'm pretty sure there are particular detectors on there that will be able to use NovaFluor dyes. As I said, we have 19 of these NovaFluors. the slotting into various different detectors in the spectral space, and so I'm pretty sure there is somewhere where in that flow cytometer where we can actually use NovaFluors. So yeah, no problem.
Yeah, absolutely. As long as you have either a blue, a yellow, green, or a red laser, you should be. There's something that you could definitely use.
I should have said that. Thanks, Anson.
This is a really good one. Do NovaFluors generally eliminate the need to do compensation? If compensation is still needed, does it need to be performed on every new batch of antibodies, just like with Tandem Dyes?
It is a good question. And actually, I mean, Anson, maybe you can help me with this, but I would always recommend to do that NovaFluors do not eliminate the need to do compensation. This is good practice. I would always recommend you need to do compensation for every dye. However, because they're a lot more stable, you can probably, how do I say this? You can probably see that NovaFluors will perform the same from batch to batch. Unlike Tandem Dyes, you don't have that huge variability. This is not to say you need to stop compensation. I would always recommend doing compensation, but you can expect a less variability with NovaFluor dyes. And so what do you think about that?
Yeah, you pretty much nailed it. So as far as compensation, that's just going to be based on the spectral properties, and it's really going to depend on your panel. If you have more than fluorophores, you're almost certainly going to need to do at least a little bit of compensation. And compensating the NovaFluors is super easy because they are so consistent. With best practices, I would probably say, at least for your first few experiments, still perform your compensation with each new batch of antibodies or each new lot. However, we have found that our lot-to-lot consistency is super, super high. Our lot-to-lot variability is less than 5%. So tandems often have a lot of variability, so you absolutely have to perform a different compensation control for each lot. With the NovaFluors, you could almost certainly get away with just doing it one time because the lots are so consistent with each other.
Yes, but I wouldn't recommend it. How does that sound? I'd always recommend, performing some kind of titration on every batch.
All right, there we go. Is there a plan to make NovaFluors excited by the violet laser?
Good question. Yes, we have plans to make NovaFluors excited. I probably didn't go into too much detail. We only have NovaFluors at the moment for the red, the blue, and the yellow-green lasers at the moment, and we're filling in the spectral spaces on those lasers as well. But we are actually working on violet and UV as well. I don't believe they're going to be released this year, but we certainly have plans to release some next year. So watch this space. How's that sound?
Very close on the violet, especially, and the UV. I think those will be coming probably early next year.
Mm-hmm.
All right, again, we've got a few more minutes, so any more questions from the audience, please feel free to drop those into the Q&A chat. Question that I have, so there's new floors coming out, and with each new floor, it seems like there's a new buffer. Do NovaFluors have any special buffers that need to be used with them, and do other buffers that are maybe not related to NovaFluors have any impact on the NovaFluors themselves?
Good question. So as I presented in the presentation, NovaFluors do require the use of a monocyte and macrophage buffer because NovaFluors bind non-specifically or can bind non-specifically to monocytes and macrophages in the same way that your PE or APC tandems do. We always recommend using the cell blocks buffer with NovaFluors to reduce this non-specific binding. What this also does is also reduce the background incredibly for NovaFluor. So I'd always recommend using that cell blocks buffer with every NovaFluor. What's easy about this is that the NovaFluor buffer, the cell blocks buffer can easily be added to, say, your antibody master mix just before you're adding this antibody master mix to your cells. Alternatively, you can also add it directly to the cells if you want to, but we've had better results adding it to the antibody master mix. Saying that, cell blocks buffer can be used in conjunction with most, if not all, buffers on the market, things like your FC block or your super bright binding buffer or your any other proteins or buffers that happen to be used on the market. We've tested most of them, not all of them, but we've tested most of them and it seemed to have absolutely no effect. So it can be easily used. The other thing is that the cell blocks buffer can be used as a replacement for your other like monocyte and macrophage buffers like the True Stain. So one final thing about cell blocks is that we include a vial of cell blocks buffer in every single NovaFluor sold to make sure that these are used with the NovaFluor. So we want to make sure that you actually get the best results out of them, and so you will have a spare buffer included with every NovaFluor.
Yeah, it's really nice that there's something that doesn't need to be purchased separately. Are there plans to offer more antibodies with NovaFluor dyes at different brightness levels?
That's a good question. I believe there is. Anson, do you want to answer that?
Yeah, that's definitely going to be one of our areas of future focus. Right now, we're really hammering hard on the violet and UV, but in the near future, I suspect we will definitely have more fluors with variable brightness.
Okay.
Would the size of phyton conjugated antibody have any problem in the detection of intracellular proteins?
Good question. I'm loving your questions, Chai. I'm sorry. Would the size of phyton conjugated antibodies have any problems in the detection of intracellular proteins? The answer is no, because in fact the size is, again, about as big as an antibody. And if you have a look at that in relation to, say, the size of, say, a PE or a PE dazzle, it's incredible. There's no problem there whatsoever. The PE or PE dazzle is going to be much bigger, and you can still use those in intracellular experiments. And so size doesn't matter. What is interesting though with intracellular work, and in fact we are currently optimizing our intracellular processes, is that there requires a little bit of blocking in order to block any non-specific binding of the phyton to any kind of DNA binding or histones that happen to be intracellular inside the cell. And so we are working on a protocol at the moment. We do have a few successes. And in fact, Anson was telling me about his successes just this morning on intracellular work, and we are currently defining the protocol for this at the moment. So watch this space. Yes, the answer to that question really is yes, NovaFluors can be used for intracellular, and we are coming up with an optimized protocol shortly.
Yeah, absolutely. The NovaFluor itself is about the size of an antibody, and the antibody fluor conjugate is still smaller than PE. So can absolutely be used for intracellular. The blocking is a little bit different from a standard intracellular protocol, and we don't have that released yet. But if you have questions about that, you can definitely shoot me an email. And I think we have time for one more question. So going back to the cell blocks that you were talking about, it's a monocyte and macrophage blocking buffer. Is it also an FC block?
No, let's just say no, it doesn't block your FC receptors, but you can use it with an FC block. So, if you have, you can use it in conjunction with your FC block. So, depending on the protocol, you can add it before or after you add the FC block. It also, as I said, can be used with a number of different other buffers as well.
Yeah, if you're looking at monocytes and macrophages, you will still need to include FC block.
Mm-hmm.
All right. Thank you, Kathryn. Do you have any final comments for our audience?
Certainly, if you wanted to look at NovaFluors in comparison to other fluorophores, there is a great table online. I probably didn't mention it in the talk, but there is a fabulous table online. If you just kind of Google NovaFluors, it comes up with the website, the Thermo Fisher website. There's a fabulous table that compares separation indices of a number of different dyes along with NovaFluors. And so you can see the relative brightness of NovaFluors in relation to other fluorophores. You can also see, again, where they fit into the spectral space as well. If you're interested in panel design, we can help you with this as well. So contact your local Thermo representative and they can assist you with this. And if you have any other questions, please, by all means, refer these to your local Thermo representatives. Thank you very much for your time. I really appreciate it. It's been lovely.
All right. Thank you again, Kathryn, for your time today and your important research. We would also like to thank LabRoots and our sponsor, Thermo Fisher Scientific, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.